Good afternoon, my name is Moníze Silva and today I will present the published results of the study Platelet gel for treating dorsal root spinal cord injury, axonal regeneration and immunomodulation developed in the Laboratório de Regeneração Nervosa at Unicamp. So, lesions that affect spinal cord roots are one of the most complicated forms of nerve injury, consisting in an important medical problem. They normally are caused from high-energy trauma, like motorcycle accidents and car crashes. This type of lesions is related with complement of ventral and dorsal roots on the spinal cord, which leads to motor and sensory loss respectively, and the formation of the glabellar scar, which affects the regeneration. In this context, the surgical root reconnections and therapies with cells, drugs, and growth factors are some of the repairing approaches and therapies for the ventral root lesions. Despite some of these therapies are also available for dorsal root contexts, the outcomes are still negative, like the emergence of an untreated pain and the permanent sensory defects, because one of the most challenges is to restore the synapses in their specific layers. Here, we try to combine some of the available therapies, aiming for a better outcome with the use of the platelet gel. The main role of our approach was developed by the platelets, a component of the blood involved in clot formation and trophic factors and cytokines release. The platelet gel that we used was a mixture of platelet-rich plasma that consists in platelets in a small volume of plasma, the PRP, with thrombin and calcium chloride. The PRP has been used in regenerative medicine since the 1990s in different contexts, also in the neural system. So, the purpose of our study was the standardization and customization of platelet gel used as an effective approach in dorsal root rhizotomy. Here, aiming to evaluate the effects of PRP gel in the dorsal root rhizotomy, Lewis rats were submitted to the lesion, and the PRP gel obtained from human blood after centrifugation process was directly applied into the injury site. After that, the animals were followed for 8 weeks. We observed that the dorsal root rhizotomy leads to 3 main events: a great cellular infiltrate mainly composed by macrophages, an extensive degeneration, and significant demyelination. We also analyzed the cellular microenvironment in the injury sites, and we found that the PRP did not exacerbate the glial reactivity 1 and 8 weeks post-lesion. On the other hand, when we analyzed the sensory synaptic circuits on the spinal cord, we found that the PRP gel preserved the glutamatergic synapses from the loss. We also analyzed the expression of several genes related to inflammation and trophic production 1 week post-lesion. Our results show that the PRP treatment has immunomodulatory properties. We also evaluated the animal sensory function during 8 weeks after lesion with the electronic von Frey test. The animals without treatment lost their sensory functions in this period. In contrast, PRP restored pawn withdrawal reflex. In addition, in the 8th week after lesion, we found regenerated axons in the treated roots. Finally, we showed that it's even possible to associate the PRP gel with cell therapy in this type of lesion, and it can be used in future studies. So, our results suggest that the repair of dorsal roots with platelet gel is efficient, and it can be considered as an alternative approach for the sensory recovery following this type of lesion. I'd like to say thank you to FAPET for the financial support, to Laboratório de Regeneração Nervosa and Unicamp, and to the organization of this Congress for the opportunity to present this project.